Hello, everyone. My name is DiIorio. I'm an MD PhD student in the Long Care Lab here at Stanford, and today I'll be presenting on a novel mouse model to study tibial distraction osteogenesis. So for a bit of introduction, distraction osteogenesis, or DO, is used to correct limb length differences and critical size defects. There are three stages of DO, latency, distraction, and consolidation. Latency is the period immediately after the installation of the device into the bone. Then there's distraction, which is the gradual period of lengthening, and then consolidation, in which the bones that have been gradually lengthened further and further apart are allowed to just sit and heal. There's several complications that can arise, such as non-union, infection, axis deviation. And because of this, we want to reduce these complications, and animal models are a great way to further study surgical techniques. So previous models are limited by expense, complications, and availability. And so we wanted to develop a reproducible mouse model of long bone DO with consistent bone regeneration so that we could further study this process. So moving into methods, here's a schematic just basically showing where the device will go on the leg. So it's on the right tibia of the mouse. This is the device that we used. It's a 3D printed plastic, two different plates that, as you can see here in our abbreviated surgical protocol, are installed into the tibia with two small screws. An osteotomy is then created in the middle here on the black dotted line, and a larger distractor screw is placed across these two plates, and the skin is closed around it. We also applied a dressing to prevent biting and degradation of the device over time. And that's basically the surgery that we performed for all of our samples. Then moving into results, this kind of just gives us a schematic of the different conditions that you're going to see in the later panels of the figure. So we had our gradual distraction group, which went through all three phases: latency, which is 5 days of sitting after we've installed the device; gradual distraction, this was done every 12 hours for 10 days at a rate of 0.15 millimeters for a total of 3 millimeters. And then consolidation was performed for 4 weeks before harvesting the bones. In our acute lengthening condition, we had a latency of 5 days before we distracted the full 3 millimeter distance all at once. And then the mice were again harvested at post-op day 43. And then in our sham control, we installed the device but did not distract, and we did not create a fracture either. So looking down at the results that came from this, this is our sham control here. We see micro-CT on the left and pentachrome staining on the right. In pentachrome, yellow is bone, red is stroma, blue is cartilage. So now looking at our next condition, this is the acute lengthening. We see a lot of stroma and cartilage, indicating a fibrous non-union. And then finally, in our DO condition down here, we see bone that looks quite similar to our sham bone. And this is also represented here when we analyze the pixel density and the bone volume over tissue volume. So this is a representation of histology and then of micro-CT. There's no significant difference between DO and sham. However, there is significant difference between acute lengthening and the other two conditions. So in conclusion, our model uses easily accessible materials and results in reproducible bone regeneration. There's significantly increased bone formation in DO compared to acute lengthening, and our DO group showed similar bone formation as sham, so that's a pretty promising result. In the past, a majority of the animal models have been done in rabbits, micropigs, dogs, and rats, and so we are trying to reestablish this in mice because it's a small animal, it's more financially feasible, and it's also genetically dissectable in a way that a lot of these other animal models don't allow for. And so we can hopefully use this model to further study DO and optimize clinical techniques. One thing we're specifically interested in looking at is the SSC, and so maybe looking at the rates of SSCs versus other skeletal progenitors at different points in the DO timeline would be an exciting next step. And so a few unanswered questions we have is, does DO recapitulate embryonic development pathways? We did sort of see that these pathways are activated in mandible distraction, but is this the same in long bone? And then another thing that's highly debated in the field is, does DO lead to intramembranous or endochondral ossification? And that's something we might be interested in answering with our model. Thank you so much for your time, and happy to chat with any of you offline with questions. Thank you.